Very glad to have with us Dr. Jody Marango, Managing Director and Senior Research Analyst with Roth Capital, who will introduce the terrain and introduce you to the rest of our panel. Thank you. Please be thinking of questions.
All right. Thank you, Bernard. And welcome to BIO this morning. I've noticed that it has become almost mandatory in scientific medical investor conferences to have a session on immuno-oncology in the last few years. So welcome to that session today. I joke it's become mandatory, but the truth is it's really secondary to how much has happened in such a short amount of time in this field, and also the fantastic clinical responses that we have seen in the field with some of the new agents. Something else I've noticed is that in immuno-oncology, there is so much nuance, and very few people actually understand and appreciate it all. Least of all research analysts like myself. So that's why we have a very nice spread range of expertise and experience here on the panel today. And to do it justice, I'm just going to let each one introduce themselves. So starting with Lou.
Hi, I'm Lou Bender. I'm CEO of Intensity Therapeutics. My background is I'm a chemical engineer, undergraduate, graduate at MIT. I worked in the material science business, and I was at a drug delivery company called Hemisphere, where I spent 14 years learning about the need for proper delivery of molecules. I spent a few years in the genomics business. I learned about cancer, and then about almost seven years ago, I founded Intensity Therapeutics. What we do is we believe the tumor microenvironment is very important for antigen recognition. So we inject our drug into the tumor directly. It's got a technology that causes diffusion and dispersion, diffusion into the cancer cells, dispersion throughout the tumor. It's two potent cytotoxic agents. When the tumor dies, it dies in a way that creates high-quality antigen. And that antigen can then be recognized. We feel, when I started the company, that the immuno-oncology field had the brakes and it had the gas, it lacked the steering wheel. So to create out of your own tumor high-quality antigen, we felt was the way to increase the efficacy of these immunocheckpoints, and it's worked very well in the clinic now. We've dosed 28 patients, and we've seen some very good abscopal effects. We don't have to inject every tumor. But we understand the tumor microenvironment is very important to getting the abscopal effect. So that's the company, and that's me.
Hi, so I'm Eric Falcon. I'm in charge of business development, licensing, M&A, alliance management at Servier. I've been in this company for quite some time, more than 20 years. I'm coming from both scientific and commercial background, and I've been doing business development for the last 10 years, helping Servier to enter in the field of oncology, and you've probably seen quite a lot of partnership that we've done in that period of time, with the focus really on both apoptosis on one hand, and the other hand is immuno-oncology. And within immuno-oncology, we like to focus on the T cells and the microenvironment around the T cells. That's really where we are working and we are most active. So I'm glad to be with you today.
Hi. I'm Sabine Kloster. I'm the Chief Medical Officer of Triumvira Immunologics. I'm a pediatric hematologist-oncologist by training out of Memorial Sloan Kettering Cancer Center and Weill Cornell Medical College. I then joined the industry, and I worked at Novartis, among other things, on the Kymriah program and helped launch the DLBCL trial, which recently got approval with Kymriah, and then worked at a small biotech, helped them launch their first cancer trial, and then worked at Merck, where I was in charge of several heme trials with pembrolizumab, then consulted briefly for a startup which tried to develop CAR T cells in solid tumors, and then joined Triumvira. So at Triumvira, we are developing engineered T cells for patients with liquid and solid tumors. It's similar to CAR T cells, but it's a distinct technology. We engage the natural T cell receptor, and we believe that we're going to have better safety than CAR T cells and efficacy in solid tumors where CAR T cells have a hard time to work.
Thank you. My name is Geert de Zerga. I'm the Vice President of Clinical Affairs at Nanology. We are a mid-stage medical development company. We have 6 INDs that are open. We have 5 INDs and 6 clinical trials. that are ongoing. I'll briefly summarize a prostate cancer study we just finished in a moment. Our approach is much like Lou's and Eric's. We're focusing on directly injecting the tumor microenvironment in solid tumors, prostate, pancreas, and intraperitoneal tumors, recurrent ovarian cancer, and so forth. And what we found, as has Lou, is elimination of the tumor leaves behind a very robust effector immune cell infiltration, B cells and T cells, that are also available for abscopal effects in other parts of the body. Our recently completed prostate cancer study, our regulatory approach, which we haven't mentioned, is actually to delay or prevent extirpative removal of the organ, in this instance the prostate, by reduction or elimination of the prostate cancer by local injection. And at least with our initial study, which, as I say, we've just recently completed, we're well on our way, and hopefully be able to follow it up with additional reports in the future. Thank you.
Thank you all. Now, we have two rather big words in the title of this panel session, immunotherapy and tumor microenvironment. It's two words, but let's pretend it's German, it's one word. Now, let's start with the second. Immunotherapies, we've heard them defined many times. In fact, ASCO and ACR and ASH have transformed themselves in the last few years around this new phenomenon. Tumor microenvironment, however, is something that we've all thought about in the past, and yet perhaps we didn't know we were thinking about it. So let's start by defining what that is, why it matters, and pardon me for framing the question and phrasing it in the following way, but let me try. When did the tumor microenvironment really first become a thing in your career? When did you first notice it as something that needs to be dealt with? Very interested in the context in that. Was it as far as school, maybe when you were 5 or 5 months ago? So very interested in the context as you answer the question as well. Lou.
Yeah, so when I started the company, I started thinking about it actually a year before. As I said, the tumor microenvironment, I thought of it as an engineer would think of it. So you can have a tumor that's a few cells. That is a different microenvironment than a tumor that's 16 centimeters. Different cancers have different environments. There's heterogeneity even within the tumor microenvironment. So when you're talking about the tumor microenvironment, you have to look at the big and the small. They may be all genetically similar, but they're very phenotypically different. So the idea was, how can you attack the big and the small, the dense and the non-dense, the lipophilic and the non-lipophilic, the hypoxic regions, the non-hypoxic regions, and the goal being to get recognition by the immune system, because if you're doing something locally like we are, you can't get to every tumor. You certainly can't even see every tumor. So I started thinking about it in terms of mass transport when I started the company from all of those variables that I just described.
And I'd like to add to that, but first getting back to your timing question, Jotun, I would have to say in my life, it would have been the early 1970s when I was at Baylor College of Medicine, and we were evaluating responses to radiation therapy to localized tumors. The TMA was not in our lexicon, but that's in fact what we were affecting, and that's where abscopal effects were actually first identified in the field of radiation oncology. And to pick up a little bit on what Lou said, in addition to all the heterogeneity and cell types, a lot of focus on T cells, to be focused on B cells and their suppressors or regulators, that heterogeneity within the tumor changes as a function of time, as a function of the size of the tumor, and as the primary tumor then metastasizes, those metastatic sites are also different in many instances. So the question of TME, is it the primary, is it metastatic, and the variability and heterogeneity of the TME as a function of time when it changes are all important considerations. And so then getting back to your IO question, we're all trying to find ways to to increase the neoantigens available using other agents administered systemically. And the problem with a lot of that, Jotun, is most of these particularly chemotherapies, are immunosuppressive. And so in a way, you're suppressing the very system that you'd like to pitch in to help with the various IO therapies. That's why I think, at least in our approach, a local treatment provides a way to think about that a little bit differently.
Well, when I started to think first about the tumor microenvironment, probably sometime in medical school, just the very basic concept that there are some aberrant cells which the body can eliminate by themselves, and there are other cells which progress and become cancer. And the tumor microenvironment is a relevant component of deciding if someone can get rid of abnormal cells, or actually if someone gets significant disease.
Yeah, I will put on another angle, probably obviously on the scientific aspect, it's quite some time, but on the commercial aspect, where really we need to focus, it's quite relatively recent in a sense that in the last, I would say, not decade, but probably 5 to 6 years, where really we always knew it was okay, but it was important to address, but we didn't have any particular knowledge of the biology which would probably lead to something meaningful in order to increase the efficacy of the chemotherapy or the targeted therapy, because there were some failures here which were, obviously explainable, but we didn't have the tool and the product in order to address that. So I would say it's more recent on the commercial aspect that it makes a very attractive, interesting place to be.
It's only fair that I mention something as well. I suppose I really thought about it for the first time, maybe not in those terms, not in those two words, but I remember when I was first seeing photos of Judah Folkman on the cover of magazines and reading about the many mice that we cured. And then since then, we've seen the VEGF inhibitors, antibodies, their performance in the clinic, etc. So I want to pivot that a little bit. VEGF inhibitors, were they the first tumor microenvironment tackling agents? And what happened there?
It's interesting that you mentioned Judah. I had the pleasure of meeting him. We were doing some work back in those days on the West Coast when he was on the East Coast in tissue repair. Not to get too far away from what we started, but we were looking at neovascular formation, trying to enhance it and suppress it in other ways. And I think Judah's view always was trying to suppress neovascularization of solid tumors. And I think he made some progress there. But getting back to Lou's point, I think the heterogeneity and the rapid changes of these, at the end of the day, the specificity of some of the VEGF preparations that he was working with, Jyotin, actually became kind of productive and frustrated him. And he was very, very successful in other areas. So I think that's where it went, ophthalmology and things like that.
So you think about the mouse models with any cancer type, especially with VEGF, and you look at curing cancer in a mouse. And when I started the company, I looked at the tumor models in mice, and you saw that with very small tumors, less than 100 cubic millimeters, and with very large drug doses, you could take an animal's tumors and eradicate it. But the toxicity levels in humans exceeded what you could tolerate at the levels of dosing that they were giving for these small tumors in mice. VEGF was the same thing. If you look at some of the early data in the VEGF, it was with very small tumors, maybe 50 cubic millimeters in the models, and then they were using 20, 30 mg per kg in the dosing, and it looked great, but then as it scaled up, it didn't work as well because the models did not really represent the human cancers. And I think that's still the case for a lot of these things. So if you're looking at data in a mouse model, just kind of go to anything, not just VEGF, you really want to look at robustness of the model in terms of size of the tumor and dose level. And if you can see for drugs that have gotten approvals and become blockbusters, they're working with something of the order of 200 cubic millimeter tumors at the start with drug doses less than 10 mg per kg. And if you look at if you differentiate by that, you can see the drugs that are going to really work. So that's what we tried to do when we started, to try to use big tumors with low dose to see if we could eradicate. And the VEGFs are a perfect example of where they don't work when you have very large-scale disease. You can't scale up. The growth kinetics of the vascular in the tumors exceed the ability safely for those VEGF inhibitors to kill the overall tumor burden.
As I think about VEGF inhibitors as perhaps the first example of agents that engage the tumor microenvironment, anti-angiogenic agents, and then coming forward all the way to immuno-oncology, checkpoint inhibitors as potentially the most famous at this point, agent that engages the microenvironment, specifically the immune component. I think about what's happened in between and how our understanding of the TME has changed. And I keep getting caught in this point, which is we don't quite, coming from what you just said, Lou, we don't quite really screen for drugs by assaying their interaction with their environment. Tumor cells, malignant cells, and then the stroma, the matrix, the supporting fibroblasts, etc. Whether it is the microenvironment or the macroenvironment, the surrounding tissue, which really there is no line there. So that's perhaps a failure, but is there a solution to this right now? Are we screening for drugs the right way so that we can come up with agents that truly affect the tumor microenvironment, short of very specific classes, like, for example, checkpoint inhibitors? Maybe this question could be appropriate when we think about the combinations of checkpoint inhibitors and other drugs. So what do you think about how we screen drugs as it pertains to coming up with something creative and good in the tumor microenvironment?
So a lot of the big cancer centers have large screening programs of their patients, and they do look at what kind of biomarkers are expressed in the biopsies or surgical specimens that they have, and then determine what kind of treatment they want to give these patients. But these kind of efforts are expensive and complicated, and it's usually limited to large medical centers. So in the vast majority of oncology patients, it's not going to be a part of these large medical centers. They are somewhere in private practice or never go on a clinical trial. So I think just getting more patients to these specialized centers or to get these tests, make these tests more available to offices, I think will go a long way in learning what can we do with these patients and what are novel biomarkers that we could target in the future.
I just want to pick up on a comment that Eric made, Jotun, in getting back to your question about what we're screening for. We started out looking at different things in different ways because of the tools we had and because of our interests. In order for biomedical science to progress, the tradition has been finding measures of commonality that we can all do and we can all agree with and work together to solve these problems. And I think what Eric mentioned has been something that's been helpful, and that is the flow cytometry identification of different types of T cells. T cells are what a lot of people are focusing on, both in academia and in industry. I would add to that B cells, and if we had another session, we could talk about B cells in the same way, or NK cells. But there are lots of different kinds of T cells, and the ratios of the T cells, the suppressors, the effectors, change as a function of time, and they change as a function of location within the TME, as Lou said. But if we're all looking at the same thing in the same way, more or less, with different tumors in different patients and in animal models, that gives us a way to assess things with a point of commonality in terms of response to intervention, as well as to the biological development of the tumors themselves.
I'm not a big fan of the way we screen for discoveries, to be honest, because I think it's somebody discovers a protein expressed on a cell, it's done in vitro, you do an in vitro screening of a bunch of molecules to knock that out. When you get into there's almost no consideration for the tumor microenvironment in the discovery process.
One postdoc, right?
One postdoc, exactly. So, one postdoc, poor guy or woman. So the tumor microenvironment isn't considered in the screening process, and then obviously when you get to the real world tumor situation, you have a delivery issue of how do you get that particular drug target to that right place in the tumor. Now, if you're doing it against the immune cells, they still have to get to the place, and you still have to get to the immune cells at the right concentration to bind to the immune cells if you're looking at that. So I think the screening process of those postdocs is a little detached from the real world problem of what we face as clinicians.
Although it's probably key to work more on the biomarkers, the identification of the population, in order to make sure that we are going to give the right drug to the right patient, and that's probably where a lot of focus should be done in the future, just to make sure. Because if, on a commercial point of view, if you put the payer's point of view, I think they would like really to have development plan who are really addressing, some population, or where you can see the tangible benefit of all this. And it's a little bit beyond the science here, but I think we should always have that in mind, that we can all engineer the T cells, and actually we can talk about it, because I think it's a lot of excitement in terms of technology, in terms of science, and certainly as a good opportunity for all of us, and for the patients certainly. But I think we should never forget that we should screen the patient.
When it comes to screening and finding a profile, biomarker or otherwise based, through which TME engaging therapies would work better, identifying those patients, we have certain capabilities right now, and potentially Sabine can tell us on the T cell side. So we're able to come up with an immunoscore and look in section, even though with immunohistochemistry you have to know what you are looking for in order to then identify it, right? We're not as good at blindly looking for biomarkers. However, as I think about it, going back to VEGF, we never really discovered true biomarkers about where it would be best used and where it would be best effective. And today, if we look at the clinical use of Avastin in oncology, I remember extension of progression-free survival, but we don't see much overall survival in advantage. So if it hasn't happened in the past, then we are counting now that it may happen with checkpoint inhibitors and the immune-oriented TME engagers. I guess, I'll throw this at you, Sabine, sort of what is the view if we just look at T cells? So, CAR Ts, TCR, the profiling of patients based on biomarkers otherwise in order to identify the right population where efficacy might be slightly higher.
So for CAR Ts, you need to look at the antigen that the tumor expresses. So you need to make sure that the target which the CAR Ts target is actually present in the patient's tumor. So that's a key biomarker. And then in terms of which patients are suitable, I think it comes more down to the overall patient, to the constitution of the patient, than specific tests which are run in a laboratory. So CAR T cells are very toxic, for example, so you need to have patients which are fit and which are able to survive the severe side effects that they will experience with CAR T cells. So these are the healthier patients, and everyone who has significant medical conditions are excluded. We don't really look at patients and say, well, we have these 10 healthy patients, they all express the right antigen, let's see now which of these would be the best. Like everyone would be eligible to get it. And we don't really understand why some patients respond better than others, besides obvious reasons that they may not express the antigen anymore, or that their immune system may not be working for whatever reason. And also CAR T cells, they work better in children than in adults. And children's immune system in general, they are more fit, the children's immune system. They can, it's able to respond better to CAR T cells. So coming back to your question, how do we look at the patients who we treat with these kind of things? It's not a lab test. It's more a clinical perspective, besides that the tumor expresses the right antigen.
And when thinking about T-cell approaches, so chimeric and TCR, and perhaps the approach that you have at Triumvira and others, they are mostly directed at the malignant cells. So they are not oriented towards the microenvironment or the adjoining cells themselves. However, they are dependent on the microenvironment. So we have seen from experiments that even though, for example, you may deliver the same amount of T cells intratumorally or systemically, you may end up with the same number of T cells in the tumor infiltrating, and yet the ones that were delivered systemically and traveled more through the microenvironment, perhaps, they are somehow losing part of their luster. They are qualitatively different once they end up inside the tumor. So framing it through the microenvironment, then, is anything being done specifically on the T cell front to somehow make them ready for what they have to travel through and penetrate the tumor?
Yeah, so there are multiple approaches. So for CAR T cells, there are fourth generation or armored CAR T cells, which are genetically modified to express cytokines such as IL-2 or IL-12, which should help them pass through the tumor microenvironment. There are other non-CAR T cell approaches. There are genetically modified T cell therapies which engage the natural T cell receptors, such as our company, and there are other companies out there who do similar approaches. And the idea is that these cells have less tonic signaling and are able to penetrate the tumor microenvironment. So there's a variety of approaches.
Yeah, Joseph, let me just pick up on a couple of these themes, but a little bit different focus. Traveling into the microenvironment, a difficult microenvironment, I'm going to refer to primary pancreatic cancer, which is surrounded by a stroma, as we all know. It's somewhat unique in the world of solid tumors. And biomarkers, I want to bring in regulatory considerations. Because I think in addition to some very exciting science and a lot of progress, people are walking out of hospitals that didn't used to walk out of hospitals because of the advances that you referred to in your opening comments. The regulatory environment has changed. Gottlieb's organization is very different right now in terms of getting things to patients broadly in phase 4 kind of scenarios and even approvals. And so using biomarkers that are clinically relevant, and I mentioned pancreatic cancer, CA19-9, CEA, those kinds of biomarkers that circulate in the plasma, we can begin to follow patients and treat patients using standard types of measures of oncologic progress by seeing their reverse, together with some sort of physical change in the tumor, a reduction, things like that. Those are now considered approvable endpoints and can allow for a lot of prolongation of high quality of life. Not so much overall survival in 5 years. There are a lot of near-term endpoints that the FDA is now beginning to take advantage of for the benefit of patients, for the benefit of industry, and I think it's a different use of biomarkers that's really helping making some major contributions here.
Just to add on the biomarkers certainly are beneficial and have helped, but it's not a binary. If you have the biomarker, you respond, and if you don't have it, you don't. PD-L1 positive patients, still there's a significant number of them that do not respond to the therapies. So they can help a lot. MSI high, yes, that seems to work very well across multiple tumor types, but again, it's not that binary of a system. There's always a search for better biomarkers, but we're not to the point yet where it's really a cutoff in a lot of cases.
Most of them are continuous variables, and it's up to us to set the thresholds, especially when speaking to the FDA, and we know how they are about thresholds, right? So yeah, that's a question. Most of the biomarkers that you've mentioned, though, are immune-oriented, most of them. Anything being done on the biomarker front for the rest of the tumor microenvironment, thinking of it as a larger population of different species, the matrix, the fibroblasts, the immune cells being a part of it. Do we have any other way to put the microscope on the rest of that population of cells and proteins, etc.?
I think it's very hard because every tumor type, not just, even in one tumor type, there's a tremendous heterogeneity of fibrin, of density, of lipid content, of size of the tumor, which makes a difference in how the biomarkers may be expressed, the necrosis levels. These are very, very complicating, big complicating factors in trying to identify a specific biomarker based on the phenotypical appearance of the tumor microenvironment itself. I'm not aware of anything that's really looking at it to a large extent because I do think it's very complicated.
That's why the fact that you have another angle to look at it. We all know the tremendous benefit that the PD-1 has brought, and you've got those 20% of patients who are really, really far better off with this medication. But it's still, there's a lot of other patients, and we need really to understand why. And that's why I think there are some, these bispecifics or this combo approach, which is now, okay, you don't have the biomarkers, but we try to understand why, a CD19, a patient is reluctant to the treatment, is resistant to the treatment, why to put a CD22 in addition so that you can really treat eventually those relapses. And this is another way you've got the biomarker way, and then the other way is, to find a complementary target in order to increase the treatment response.
I will say that there is some research in this field at USC where they're looking at radiographic imaging and using PET scanning and some other very, very exotic imaging techniques to look at the density of the tumors that can show up in fine detail across the tumor to try to distinguish which patients may be more responsive to certain types of therapies.
Yeah, there are novel mechanisms such as single-cell sequencing, imaging, flow cytometry, but the bottom line is we don't really understand the tumor microenvironment. I mean, we have all these techniques and we have all these models and some data out of certain clinical trials, but overall, we don't know. So I think that we need to move towards a way that we actually learn more and that we can combine the data from these various trials and look at large data sets. And then from looking at these large data sets, hopefully we can come up with novel ideas. What are our next therapies that we want to develop, or what would work?
Is there, asking the last question in a different way then, since most of our therapies so far are tackling the immune component of the microenvironment, T cells, macrophages, NK cells here and there, is there, so rather than biomarkers, any approach that you know of that is tackling the rest of the microenvironment that is non-immune in nature?
With that kind of a challenging question, Jotun, I'll give you a curveball out of left field answer. We've done some work in orthotopic non-small cell lung cancer models with a variety of cell types. So these are all animal studies. And what we found is that we can eradicate the tumors in the lungs with inhaled these submicron particles. And what we find in the lungs that no longer have tumor is we find very small plaques of fibroblasts. And so that has led us to wonder if these are tumor-associated fibroblasts, which are part of the microenvironment that really haven't been studied. And we're looking at some genetic mapping to see if in fact they are, in contrast to just a replacement fibroblastic response as might occur with a wound. And I think it's going to be the former more so than the latter. Another type of observation that we've made in a number of different biological models that does occur in humans, particularly in lung cancer, are what are called tertiary lymphoid organs. They're collections, oftentimes of B cells, sometimes T cells. They're activated. If you do immunospot analysis, they produce cytokines, they produce antibodies. And so it's, not to add more complexity to an already complex situation, but the biological organization of the individual or the animal model in response to the persistence of therapies also changes the game. So if you go back to the intravenous therapies versus long-term local therapies with the same cytotoxic, you get completely different responses based on the tumor's response and based on the host's response to that duration of cytotoxic exposure.
Okay, so interesting, and neither have I, and I think that that's a real challenge, trying to tackle the microenvironment beyond just the immune component. And as we said, that also comes as a result of what we can do in terms of biomarker screening. Most of the biomarkers we report on and we're involved are around the immune system. That's fair. I'd like to stay with some of the questions that we've received from the audience and maybe transition from CAR-Ts, and you'll see, perhaps a conceptual pivot onto bispecifics as another way of engaging a component, the non-tumor component. So I was going to actually direct this at Eric, given that at his organization they have done, there's been a fair amount of activity around bispecifics. So what are your thoughts about bispecifics, and is there a component, how do you think of them as engagers of the microenvironment, perhaps?
Well, again, clearly I think we have a lot of activities with bispecifics, whether there are standardized bispecifics or standard ones, or whether combination of immune checkpoints. I think we've got the two type of activities at the moment. I think the way we view that was, again, we put the payer's point of view, the patient's point of view, and obviously if you are going to develop very expensive therapies with different type of targets, and I think you want to address, with a mechanism which makes sense, with synergies which make sense, you want to address that in one single drug, one single monoclonal, whatever, bispecific you want to address. And this is the angle we've taken. I think we believe that it's not just one immune checkpoint who's going to work. I think you need to combine those in order to increase the response rate. Clearly, in terms of the same for the CAR-T, I think we believe the combination, although it's extremely complex and on the scale of risk taken, I think you are moving in a high level of risk there. But I think that would be an answer for the patient in order to have a simplified way of taking the drugs, and it should be synergistic. So there's a lot of basic research, what is the right combo, what you want to enhance, and all these activities done on various type of answers, but no, apart from one, not really a lot of drug has been already approved or in this field. They are all in development. So we are really in the high risk and still speculative way of doing, but very promising.
Yeah, I think you complicate the matter very, very dramatically if you're trying to take one molecule to bind to two different sites in a real live human being, where you've got a large heterogeneity of the tumor microenvironment again, or the person's physical immune heterogeneity between patients. Now you've trying, and remember, from again, chemical engineering, binding is proportionate to concentrations, and those concentrations of those proteins and those and the antibodies can change, and their binding kinetics can change within the environment. The pH makes a big difference. How those 3D structures are being presented at the receptors. The receptors can be different under different conditions. So you now have, with antibodies already a complexity, a single binding antibody. Now you've got a bispecific antibody into two very different sites where you have very different environments for each of the binding kinetics necessary to make that bispecific function. So it's the hot thing now, I understand that, but I think it's got a rapid cooling potential given the complexity level that exists with two binding sites in one molecule.
That's funny. I was actually hoping for I was expecting a different answer there, because I was, moving. Fantastic. I was moving from the CAR-Ts. You're handling cells with their degree of sophistication. They have a mind of their own, onto something that is potentially perceived, at least by many physicians as well as investors, as a simplified version of an engineered T cell. If anything, from a tumor microenvironment standpoint, perhaps in a cartoonish way, it might even be somewhat more versatile. It might penetrate the tumor better, just as an antibody. And yet, you were saying that the same challenges would apply, if anything, potentially more. Do you have any thoughts on this, Gary?
I was just going to add, I'm surprised, Jotun, that you were expecting this is less complex, because I think Lou's right on with what he's saying. And in addition, it's the retention of the conformation of these very complex molecules to identify biologically adaptive sites in a conjoined way. So it makes a great deal of sense, but the application for an IV administration to get through the body into the tumor microenvironment to identify these cells, the immense challenges. It brings me back, you mentioned Judith Faulkner, it brings me back to the early days of ELISAs, when we were working on stationary platforms and trying to get bidirectional identification. It's difficult to do, and now we're trying to do it intravenously as a delivery system into the TME, which doesn't particularly want to be penetrated.
Very fair. And we know that as the tumor progresses, and I think you alluded to this, the tumor microenvironment is different between different sites in the body, but also temporally. It actually changes, right? It becomes more rigid and more solid as the tumor progresses. Exactly. And so, it lets things out, metastasis. It does not let things in, cells, antibodies, and therapy.
Well, actually, I've got just a little bit of, it was a surprise to me, hopefully not my fellow panelists or anybody else in this room. We're doing work with the humanized mouse models, and the response of the intravenous antibody therapies changes dramatically based on the size of the tumor that you actually treated. Lou alluded to this a little bit earlier, and so that's part of it too. And my guess is how long that primary and that metastatic lesion has been in a patient's biological system is going to be just as important as the duration of time and the size of the tumors that are well characterized in these humanized mouse models, because we're using the human immune system.
Well,
yeah, well, I would like to add on the bispecific, but especially on the CAR T, that's clearly in AIM, it's making sense, or at least we can go forward with all the question you put on the complexity, and for solid tumors, we are miles away here. I mean, we clearly don't understand the consequence and all the biology around all this, and that's a step too far, I think.
I will agree that transport in for a bispecific antibody is going to be easier than a cell, depending on the tumor environment, especially in very complicated large tumors. In hypoxic regions as well. But it adds a degree of complexity for functionality, not as much for transport, but more for functionality.
very fair. And to be even fairer, we could have had a separate panel discussing all of these therapies and the microenvironment in liquid tumors, in the bone marrow, hematology, and one on solid tumors, which is, as you said, miles behind. But somehow we have, mixed the two here and there. I want to take a few questions, if there are any, before we actually continue with our own questions here. So I just want to open the microphones in the audience, if there's any questions. Yes.
My name is Charlie Craig. I'm with Opus Biotech Communications, and my question is, what role can these new therapies play in treating certain local tumors to prevent disease progression and removal of organs such as the prostate, the bladder, and the kidney?
So clearly, obviously, local disease is what you can see and not what it may actually be, right? So if you have a very large tumor and it looks like a single tumor, right now the standard of care is cut it out and then bathe the person in some sort of cytotoxic chemotherapeutic agent, especially if they're node positive. That is the paradigm that works very well for the majority of cases. There will be cases where that's inoperable for some reason or another, such as in pancreatic cancer, where it's often very challenging to operate. There's only 20% of those tumors can be operated on. So I think in certain tumor types, you'll have a local therapies like a radiation or ablation, but those have limitations such as where that they can't be done greater than 3 to 4 centimeters in diameter. So I do think there's a need for a new type of local approach for single tumor types that are inoperable, where there may be metastases or single cells that are being shed that you can't see. And that could be a very big market for people because there's a lot of those cases that exist today.
Actually, let me just add, you mentioned bladder, you mentioned prostate. There's actually data available on, and Justin, I think it's overall survival. It's certainly progression-free survival on individuals, as Lou said, who had not undergone prostatectomy or cystectomy, who had muscle-invasive bladder cancer and prostate cancer that had some extracapular extension and maybe some metastatic disease to the nodes. That suggests, at least, that if the primary is treated with some retention of the primary, and then the patient goes on to receive intravenous IO therapy or chemotherapy or both, that actually there's a benefit. And the idea is that there's something about treating the primary disease, but leaving the organ in place instead of what we were all taught in medical school, take it out. Leave it in place for a certain period of time, receive the intravenous therapy, and then do your extirpative surgery. So there actually is data available in both of those areas that would suggest that there's a patient benefit.
And it's early days. I mean, everything is a risk-benefit ratio, and it's a balance between efficacy and safety. And the field is really young, and everyone, or a lot of people, are working on novel therapies, on more efficacy, better safety. And if you have a therapy which is efficacious and has no safety problems, you can also use them in local tumors. But the way we do drug development is we usually treat patients first who don't have other options. And then once we have the safety and efficacy profile established and see if it's better than existing therapies, then we move it to earlier lines of therapies. And potentially all these therapies we are speaking about right now could be frontline in a couple of years from now.
And Gear, what you mentioned, I find that we've heard versions of that from the, I was going to say the older days of chemotherapy, but it's still around and I don't think it's going anywhere. But even that, we thought that once we thought it was immunologically silent, but it isn't, right?
And remember, even though the history of therapeutics and disease has this timeline, the human condition hasn't changed. It's just that we now are better able to recognize what an important role the human in both adaptive and inherent immune systems play in these things. When you and I were in medical school, that was not the case, or at least when I was in medical school.
With chemo itself, but also some of the targeted agents, oncogene targeting, we've also thought that apoptosis was immunologically silent. It isn't. So even taking chemo, shall I say, I don't know, this is on the record, I shouldn't use terms like this, but perhaps at low doses, it still can have an effect on the tumor via the leakage of proteins, etc., that are immune activating. So again, now, chemotherapy itself is immunosuppressive. So, there's a fine balance there, but there's interesting work that shows that the lower dose use of some of these older agents, the cytotoxics, is actually quite immunostimulatory and productive.
And almost counterintuitive, the low doses in terms of the high dose intravenous, you almost get the opposite effects in terms of the cellular response, the tumor cellular response. And the other thing I would add to that, which we wandered into unexpectedly, is that the persistence, the continued exposure of tumors in situ over weeks instead of a few hours as occurs intravenously changes the ballgame entirely. There's a very different response because the T cells are modulated, the exhaustion issues, the suppression issues, they respond to differences in persistence of exposure to chemotherapy.
And there are new ways of delivery of those chemos now right to the tumors, which has also evolved and gives a better efficacy.
And to your point, cisplatin is well published that it stresses the endoplasmic reticulum, releasing calreticulin to the surface, attracting antigen-presenting cells. The platinum agents do that very strongly, and it's been well documented for 20 years.
Yes, but I still want to be forward-looking, so we'll write off the last few minutes to nostalgia and fundamental science. So immuno-oncology, please.
Hi, good morning. I'm the president of the Children's Tumor Foundation, so of course I was very enthusiastic to hear that you said that these T-cell therapies could work better for pediatric patients. So my question is, when you say, Sabine, that you think that immunotherapies today are not yet first-line treatment, but may become it, could the panel a little bit speculate on what it is that we have to do in order to get to that point? I mean, sorry, this is a $5 billion question, but I think it's kind of an interesting speculation.
I actually think you probably heard about the success of Kymriah, which works beautifully in relapsed refractory ALL. And I think the Children's Oncology Group is starting a trial to evaluate that frontline because a lot of parents put pressure on the group. And it's, I think it's a matter of time. As more as we learn about these therapies, and assuming that as more as we learn how to manage their safety profile or how to come up with technologies that prevent the safety problems which we are seeing right now, these therapies will move frontline.
Well, I would add something which is very important is the CMC aspect of the process to get those products, especially for the CAR T. You know, the process gives you the drug. You know that a good process can have versus a bad process can really give another level of efficacy and safety. And in order, if you really want to move first line and front line, you need to be more cost effective. So you need to be able to produce those at a lower cost because obviously affordability is going to be, for a wider population, is going to be an issue. So I think the big work that has to be done now is really to make sure that we can really produce those medications at a price which is really affordable in order to be able to sell it at a good one. So that's really a key element, CMC.
So if I may circle back, Lou and Gear, see bispecifics are at least vindicated in one component of their, yeah.
There's a few questions that came in via the bit.ly link, so I'm just going to read them off and let you all determine which ones to answer because we only have a couple of minutes left. The first one was what degree of hope in neoantigen-based therapeutic, what do you think the degree of hope in neoantigen-based therapeutic cancer vaccines, the status of dendritic cell-based approaches, and then the tumor microenvironment necessitates a single-cell analysis, complex assessment of millions of single cells for companion DX, TX, what tech are you using? And there were a few examples, mission, bio-DNA, myeloprotein.
I'll take the vaccine, so I don't have to talk about the other ones. Thank you for your question. I think the vaccine story is something that doesn't receive frontline attention, but I think it will be a tremendous attribute that comes out of the current therapies that we're talking about and that you folks have read about. We're generating therapies in a variety of ways that at the end of the day produce cells that have memories. We've all talked about abscopal effects. We've all talked about B cells and T cells responding to various manipulations. Most of those cells will have memories. And with memory cells, it's very easy to move into vaccines. Hopefully the vaccines aren't specific to individual patients' antigens. Hopefully they're more general than that. At least the animal studies suggest they are. There's a great deal of work going on in GU, in pulmonary diseases, that I'm aware of. And the vaccine stories, I think, will be something that we will all live through. It will take a long time to demonstrate their societal benefits, as is the nature of vaccines, but I think it's part of the benefit of the approaches that we're all taking.
I'll highlight again the first question, which was, I think, what is being done to make the tumors hotter, as I understood it, so to increase their complexity and neoantigens as a way to increase efficacy of these approaches that we have on the table. So, and I'll be curious to actually hear what Eric has to say about this, given his position and the fact that he looks at so much every day. So yeah, what do you know on this front?
Well, I think there's a lot of work to be done. I think we can spend hours on this, on neoantigen. I think we need to discover, especially for solid tumors, there's a lot of basic research, basic work that has to be done in order to find new antigens. I think we don't know very much. A lot of those to address solid tumors, that's for sure. In a more general way, and on the vaccine as well, because I really wanted to say something that this is very exciting. But if you believe that bispecific and CAR-T are complicated, vaccines, oncolytic, it's another level of complexity as well. So it's not going to be for now; it's going to be for the future. But I think it will happen. That's for sure.
Lou, did you?
Well, there's a lot of intratumoral approaches with STING and RIG1 and trying to get toll-like receptor binding. That's being done. The data's early. It's still very young in the field. So I'm hopeful that they can, that there can be some way of turning other than what we think we're doing on it, to turn a cold tumor hot. We'll have to see. There is a lot of different things going on
within the tumor microenvironment.
In the tumor
hoping that it does not cool it down once we heat it up.
Yeah.
Any last thoughts about the general area of tumor microenvironment? We should wrap up in a minute or so.
Well, I just say I think there isn't a lot of focus and funding of that in academia. I'd like to see maybe the NIH start to have some more R01 grants to fund tumor microenvironment research. I don't think that's being looked at significantly.
Yes, Sabine mentioned that actually a lot of work has to be done. We need to know better what's going on there, the biology and the like.
And I think we need to collaborate more. I think sharing data sets and just analyzing data across different companies, different continents, different indications, I think is something we need to do in the future and will give us a lot of information of how we can tackle cancer better.
And I think the FDA recognizes this, and as in this country, FDA defines the truth from a regulatory perspective. The FDA is having meetings, open national meetings with invited speakers to TME and other specific areas. They're well attended, they're available publicly, and I think they are setting the stage for collaboration. They are setting the stage for additional federal funding, and I think it's something that we can all get together at the same time, compare notes, and try to come out with a better product at the end.
Very good, and I'm glad that, Geir, you brought up the FDA earlier on and how lenient and open it's become to a biomarker-based approach to assessing efficacy and developing drugs. Because at the end of the day, I think it was a CEO at Bio this time last year that joked with me and he said, "Who is your customer as a biotech company? It's actually not the patient, it's the FDA." But hopefully we're all moving forward in the right direction. Thank you very much, Geir, Sabine, Lou, and Eric. And please find them afterwards. They have encyclopedias in their heads. And thank you for coming. Have a productive meeting.